One of the questions about any type of gene regulation or any type of event is to ask whether or not it's conserved. So as Dobzhansky was saying, nothing in biology makes sense except in the light of evolution. So presumably if something had been going on since our long gone ancestors, since like tens or hundreds of billions of years ago, then presumably there is some reason for that to happen. So it didn't dissipate in the in all that time. We found that in humans there is a large fraction of genes, so between a few hundred to a few thousand, can be expressed from one copy or from the other copy, from one allele or from the other allele. But it's not, or it wasn't completely clear whether or not it's the same set of genes that are in the mouse or not. And among other things, even apart from the sort of lofty evolutionary questions, if you know what's going on in the mouse, it's much easier to manipulate genetically. So you can get tissues that you cannot easily get in humans. You could do transgenic mice, you could do all kinds of experimentation that is either impossible or very difficult in if you're working with human cells or tissues. So what we did, we took clonal cells from the mouse, and these were specific mice. We crossed them, so mostly people work with different lines of mice, and what they want to do is to have inbred mice, so that there is as little variation between mice as possible. So you can have a colony and all of them are genetically identical. So what we want is we want to separately measure maternal copy and paternal copy, and in order to do that, we want to have differences between them, and the differences are sort of colloquially known as SNPs, which is single nucleotide polymorphism. So we crossed one standard lab strain, 129, with a wild type strain called castaneus. So on average, what happens is that about every 124 bases, there is a difference between these mice. So their progeny is perfectly fine. They have some trouble mating, but they're sort of happy mice. And you could look at every tissue, and you could look at them, and you could see what the difference is. So here is one gene, here is another gene, and there is a T, there is a G, and based on this SNP, you could measure relative levels of expression of a gene. So what we did then, we took B cells, or actually pre-B cells, and made clones of them. So we used FACS sorter in order to do, you place a single cell in a well, then it expands, so you have, so cells divide, and you have something like 100 million cells. And then it turns out that if you take RNA from these guys, and then you use a different technology that was developed for sort of different purposes, so-called SNP chips. They were developed to genotype, to look at the genomic DNA and see whether, what in a given position, what the nucleotide is. So for example, if you look at one parent, 129, it will be whatever it is, like T here. If you look at another parent, it will be homozygous G in that position. And if you look at this F1 cross, then it will be heterozygous TG. So you genotype father, you genotype mother, and they're homozygous for T, homozygous for G, and when you look at the F1 cross, what you see is it's heterozygous for T or G. So that's what this technology is for. So we sort of subverted it to measure what's going on with the RNA or other cDNA, and asked what would be the bias. And it turns out that if you take clones from, even from the same mouse or the same type of cells from individual mice, it turns out that the differences are pretty big. So if you take cells from females, so one obvious difference is that X chromosome will choose one or the other direction to be inactivated. But also in a lot of genes on the autosomes will choose to be expressed from this allele from the paternal allele here and from the maternal allele here in a non-imprinted way. So some cells are choosing this, other cells from the same genotype, even from the same individual, are choosing something else. So one thing was that the fraction of genes was pretty similar to what's going on in humans, which is good. So 10–15% of genes of those genes that we were able to measure, which is a couple of thousand genes, were expressed in this manner, sort of autosomal analog of X-inactivation. What is more interesting, so we looked at the same cell type as we previously looked at the humans. So very similar genes were affected. So that means that, basically, if you know what's going on in the mouse, it's very likely that the same gene is affected in people and vice versa. And it also means that whatever is the mechanism that tells that this gene does this and this other gene never does something like this, this mechanism or whatever persisted since we and mice had last common ancestor. Another interesting thing that we didn't expect was that there is another potentially deep analogy with X-chromosome inactivation. So what happens usually is that about half the cells decide in one direction, half the cells decide in another direction. So when you look, if you take the whole mouse, you put it in a blender, look at what's going on with expression, then both copies of the X are on. It's just that each individual cell is 100% chooses one copy or 100% chooses the other copy, and the other copy is completely silenced. But if you look, for example, at the same couple of mice, 129 and castaneus, when you look on average, what happens is that you have something like 80–20 skewing towards castaneus allele. And it's not because in each individual cell, one allele is somewhat expressed and the other allele is expressed much more. So what happens is that in each individual cell, it's still all or nothing thing. But out of each 100 cells, 80 cells would choose castaneus allele and 20 cells would choose 129 allele. And then everything goes on as it went before. So we see something quite similar, where we look, for example, at 10 individual clones. So we do the transcriptome-wide analysis. And what happens is that you see that 8 or 9 clones have chosen one direction and 1 or 2 clones have chosen the other direction. And there are way more of these genes than you would expect by chance, just from sampling error. So it seems like this is similar to what is going on with the X-inactivation. It's known what the mechanisms are to pretty great detail. So there is a genetic element on the X chromosome called X-inactivation center, and depending on precise sequence of it, it determines sort of the strength of the X-inactivation center. And that's why when you cross castaneus mouse with 129 mouse or other lab mouse strains, it's more likely, the choice is more likely to occur in one direction than in the other direction. So this leads us, so a similar observation on the autosomes leads us to a hypothesis that there is something very similar. So there is a genetic element that controls this epigenetic switch early in the development, and that means that we should be able to narrow down and find it and live happily ever after. Sort of major problem that is there is what is potentially this regulatory elements. Finding them, so it's presumably some non-coding element, which sort of we hypothesize for a number of reasons that it's probably fairly close to the genes that are affected. And that means that among other things, we would be able to do genetic manipulation of this. So if we're unhappy about a certain gene behaving in this manner, we would be able to sort of reset this and see what's going on. But one thing that we need for this is a very high density map, and ideally a very high density map of allele-specific expression in various tissues. And there are major challenges there. So one major challenge is that it's very difficult to do, so this type of behavior is observable in clones, but it's very difficult to obtain clones from a lot of different tissues. Even more challenging is that oftentimes cloning is happening in culture, in cell culture. That is not in vivo situation, so maybe we're introducing artifacts by doing this. So we think we now have solved this issue, and we now can look at this, but that's subject to another story, so which I'm not going to tell about. So there are definitely plenty of questions there, but the availability of the mice means that we can now do genetic manipulation, that we can now compare different strains of the mice, and we can build these extremely high density maps and see what's going on. And now a range of new technologies came in with the high throughput sequencing, and we're building tools to do that type of analysis, which will allow us to figure out what is genetics of this epigenetic mechanism.